Ever wondered how scientists edit genomes? It all began with a breakthrough in our understanding of life's blueprint, DNA. Picture this. It's the early 1950s, and the scientific community is abuzz with the discovery of the double helical structure of DNA. This structure, a winding ladder of genetic information, was a revelation. It unlocked the secret language of life, allowing us to read, interpret, and eventually manipulate DNA sequences. Yet, as with any complex language, it posed challenges. How to precisely change DNA sequences at exact locations. How to overcome the inherent complexity and variability of genetic code. These challenges were daunting, formidable. But as we know, science thrives on challenges. This discovery of DNA's double helical structure, as complex as it was, didn't deter researchers. Instead, it fueled their curiosity, their drive for knowledge. This discovery paved the way for advancements in the field of genetic research. From early techniques to modern marvels, the journey of genome editing is fascinating. Indeed, ever since we unraveled the double helical structure of DNA, we've been on a quest to manipulate these sequences. But, let's be honest, creating precise sequence changes at specific sites has been a tough and meticulous undertaking. Our initial attempts were somewhat successful. We used oligonucleotides, small molecules, or self-splicing introns, each approach with its own set of challenges. We were like explorers in a vast genetic landscape, finding our way with rudimentary tools. Then came the era of zinc finger nucleases, or ZFNs, and TAL effector nucleases, also known as TALENs. These were our first sophisticated tools for sequence-specific manipulations. Think of them as our genetic GPS, guiding us to the correct location in the genome. Yet, even these had their limitations. The design, synthesis, and validation of these engineered nucleases were no simple task. It was like trying to find your way in a dense forest with a detailed but challenging to decipher map. These techniques, although promising, were laborious and time-consuming. They were like early steam engines, powerful but complex, and not particularly user-friendly. They set the stage, however, for a scientific revolution that would change the way we interact with the genetic code. This revolution came in the form of the CRISPR-Cas9 system, a technique that has largely overcome the difficulties of previous genome editing methods. It's like moving from a steam engine to a high-speed bullet train. The CRISPR-Cas9 system has proven to be so straightforward and economical that it has been likened to the democratization of gene targeting. So imagine the evolution of genome editing as a journey. We started with basic instruments, navigated through complex techniques, and finally arrived at a place where precise, affordable, and accessible genetic manipulation is possible. Though these techniques had limitations, they set the stage for the emergence of a transformative technology, the CRISPR-Cas9 system. And this, my friends, is where our journey into the future of genome editing begins. Join us as we delve into the rise of this revolutionary technique and its impact on the scientific world. Enter CRISPR-Cas9, the game changer in genome editing. Born from the humble bacterial adaptive immune system, this technology has the potential to revolutionize our approach to genome editing. But how does it work? Well, it's a two-component system. The first is an enzyme called Cas9, which acts like a pair of molecular scissors, snipping double-stranded DNA at precise locations. The second is a guide RNA, or gRNA, that acts like a GPS, directing the Cas9 enzyme to the correct location in the genome. Without a repair template, the DNA break caused by the Cas9 enzyme is repaired by a process known as non-homologous end joining, which is somewhat prone to errors. This can result in a mixed bag of cells, each with different insertions or deletions near the break site. However, this can be used to our advantage to create cell lines with a specific gene knocked out. But what if we want more control over the sequence changes? That's where a repair sequence comes in. If provided, the DNA can be repaired using this sequence as a template, allowing us to introduce precise changes at a specific location within the genome. This gives us an unprecedented level of control over genome editing. But it's not just the precision that makes CRISPR-Cas9 a game changer. It's also its simplicity and cost-effectiveness. Unlike previous genome editing tools, which required complex protein design, synthesis, and validation, CRISPR-Cas9 is easy to use and inexpensive. This has led some to suggest that it could democratize gene targeting, making it accessible to more researchers and potentially transforming the field of genetic research. CRISPR-Cas9 has revolutionized genome editing, but to fully utilize its potential, we need efficient screening methods. This is where Sanger sequencing comes into play. Stay tuned to find out how this tried and tested method can help us make the most of the incredible potential of CRISPR-Cas9. To ensure precise genome editing, we turn to a trusted ally, Sanger sequencing. Embarking on the journey of genome editing, we've seen how crucial screening and identifying the right edits are. This is where Sanger sequencing steps into the spotlight. It's not just a tool, but rather a guiding hand in this intricate process. Imagine this. You have a pool of transformed cells, but you need to find the ones with the precise alterations you introduced. How do you do that? You could look through a microscope, but even the sharpest eye can't see the individual genetic changes. This is where Sanger sequencing comes in. It's like a fine-toothed comb, meticulously going through the genetic material to identify the changes you've made. What makes Sanger sequencing so special? Well, it's simple, cost-effective, and straightforward to analyze. In a world where genome editing techniques can be complex and expensive, Sanger sequencing is refreshingly accessible. It's like having a reliable old friend who's always there when you need them. But the benefits don't stop there. Sanger sequencing has another trick up its sleeve. It can identify sequence changes and detect mixed single nucleotide polymorphisms, or SNPs, in a population. This is a real game changer. Imagine being able to spot not just one, but multiple changes in the genetic material. It's like having a superpower that allows you to see the smallest details, making sure nothing slips past unnoticed. So as we dive deeper into the world of genome editing, we find ourselves turning again and again to Sanger sequencing. It's not just a tool, but a trusted companion on this journey. It ensures that our edits are precise and that we can confidently move forward in our research. In the realm of CRISPR-Cas9 workflow, Sanger sequencing stands tall. It's not just about making changes to the genome. It's about making sure those changes are exactly what we intended. Sanger sequencing, therefore, plays a pivotal role in optimizing the CRISPR-Cas9 workflow, ensuring the precision and efficiency of genome editing. The future of genome editing is promising. With advancements in technologies like CRISPR-Cas9, the potential for precise, cost-effective genetic modifications is expanding. Yet, the role of Sanger sequencing remains crucial, providing reliable and unambiguous data. Looking ahead, we can anticipate the development of more sophisticated tools and techniques, enhancing our ability to manipulate and understand the genome. As we continue to unravel the mysteries of the genome, the combination of CRISPR-Cas9 and Sanger sequencing will remain at the forefront of this exciting scientific frontier.